Hello to everyone. Welcome to EPG Pathashala program in botany subject. I will be talking on a topic from plant physiology module, that is mechanism of cell expansion and cell elongation in plants. Plant growth and development has three developmental processes, that is cell division, cell elongation and cell differentiation. These three developmental processes has key mechanical aspect that has prompted various scientists to develop theoretical, mechanical and physical models. Let us first begin with the cell expansion. Cell expansion has two key steps. The first one is increase in the cell volume and the other one is a change in a cell shape. Increase in the cell volume from a meristematic cells to a differentiated plant cell type is essentially from 10-fold to 1000-fold, but in some cases it could be, for example in case of xylem, it could be 30,000-fold. Looking at variety of such differentiated plant cell types, the additional cell wall that has to be generated, that has to be created to develop elongated differentiated plant cell is very impressive, and therefore an understanding of a mechanism for plant cell expansion and plant cell elongation in plants is essential. In this module we will study the learning objective of mechanism of plant cell expansion and cell elongation. These are: study the shoot apical and root apical meristem and the overview of cell growth, study the growing plant cell wall, study cell expansion, study the role of expansin in cell expansion, study cell elongation, study the role of cellulose in cell elongation, study the cessation of growth, that is, how does growth stop. The shoot apical meristem is a small population of cells that continuously generates plant organs and tissues. The shoot apical meristem is highly organized structure, and a simple histological analysis is sufficient to distinguish between different zones and layers of a meristem. At the meristematic dome lies a so-called central zone where a small population of cells assures the maintenance of meristems, and this central zone is surrounded by a peripheral zone where the organ primordia are initiated. Roots also display a well-defined developmental gradient along their longitudinal axis, with young cells being close to the root tip and the older cells at the root base. The root apical meristem, located close to the root tip in the meristematic zone of the roots. Here, a single layer of a cells is maintained by four central cells, the quiescent cells. The stem cells distal to quiescent form and continuously renew the protective root cap, while the stem cells proximal to the quiescent cell produce a main root body. New cells are displaced from the quiescent area will no longer to be maintained in a stem cell state, and they enter into a transition zone where the cells divide rapidly and produce more cells. Once they pass the transition zone, the root cells will cease to divide and enter the elongation zone. Here, the cells elongate until they reach their final size and obtain specific functions in the differentiated zone. In a growing cell, turgor pressure generally remain constant. Cell expansion as well as cell enlargement must be the result of controlled expansion of cell wall. The growing plant cell wall is a polymeric structure consisting of crystalline cellulosic microfibrils coated with the hemicellulose chains, which are embedded in highly hydrated pectin matrix with small amount of proteins. In growing cells, these microfibrils are laid down transversely to the axis of elongation and thus forming a spring-like structure. This spring-like structure reinforces the cells laterally and favoring longitudinal expansion. Hemicellulose and pectin are synthesized in Golgi apparatus, transported in vesicle to the cell surface. Upon the secretion into the apoplast, hemicellulose bind tightly to the cellulose and are thought to link to the microfibrils in a stress-resistant network. Cell expansion requires the constant rearrangement of bonds within this stress-resistant network to allow the movement of wall polymers and the incorporation of new material into existing architecture. Cell expansion is an essential step in determining the final leaf size as well as the root or any other organ size that is governed by different mechanisms in each stage of cellular development. Expansion in meristematic cells is determined by both increase in the cytoplasmic and nuclear volume, whereas expansion in differentiated tissues, it is mainly determined by turgor-driven vacuolar enlargement that allows accumulation of water and solutes. Cell expansion is a result of vacuolar enlargement as well as turgor-driven cell wall yielding. The vacuola expand while take up water and solutes, whereas turgor-driven cell wall yielding is a result of multiple steps including hydration and cell wall loosening. Cell wall extension by turgor pressure dehydration or cell wall stiffening by release of apoplastic reactive oxygen species and cross-linking and dehydration, as well as synthesis and accumulation of cell wall components. Cell wall loosening is controlled by expansin proteins and xyloglucone endohydrolases and xyloglucone endotransglucosylase activities. Cell wall relaxation stretches the plasma membrane, which promotes the opening of calcium channels. The resulting increase in a cytoplasmic calcium affects the growth by inhibiting phosphorus dependent ATPase enzyme activity that causes alkalinization of apoplast and inhibition of expansin activity. It also activates NADPH dependent oxidase, which promotes secretion of superoxide into the cell wall, which further convert into hydrogen peroxide. These reactive oxygen species promote cross-linking of cell wall component, which causes cell dehydration and cell wall strengthening. Growing plant cell walls typically extend at a faster rate at a low pH, a phenomena referred as a acid growth theory. This is easily seen in isolated walls clamped at constant tension in extensometer that you can see in a figure number 1. At neutral pH, the walls soon stop extending, but they rapidly extend when the pH is lowered. Acid-induced extension is not merely a physical property of wall polysaccharides, but it requires active wall proteins. A key breakthrough came with the discovery that wall proteins may be added back to the denatured cell wall to restore their ability to extend. The active proteins were of new protein class, subsequently named as expansin. Their action is referred to as a wall loosening, a general term that does not specify exact mechanism of induction of cell wall expansion. Expansins are those proteins with a relative molecular mass of about 26,000 were isolated for the first time from young cucumber seedlings and from subsequently from other plant tissues where their activity was associated with cell growth. Expansin was also expressed during the drought responses of maize seedling, whereas maintenance of root growth involved increased expansion activity in a growing region. Recent work indicates that drought also increases expression of expansin gene in spatial and temporal pattern that closely matches with the changes in the expansin protein activity. Let us begin with cell elongation. In the elongation zone, cells rapidly expand along the longitudinal axis via cellulosic loosening. In the elongation process, cells prevent lateral expansion of cells. Cell elongation is primarily achieved by cell wall enzymes which remodel and loosen the cell wall, thus allow for cell wall extension during increase in turgor pressure. During cell elongation, auxin promotes acidification of cell wall, which is now also called as acid growth hypothesis. Here in this process, protein pumps activated by auxin, and it leads to acidification of wall that trigger the activity of expansin protein. Expansin also loosen the cell wall at cellulose xyloglucone junction, allowing for slippage of cellulosic microfibrils. This activity leads to a extension of cell wall and leads to a further creation of elongated cells. Let us discuss the role of cellulose in cell wall elongation. Cell wall is generally 0.2 micrometer thick and completely coats outside the plasma membrane. Cell walls has layers of cellulosic microfibers embedded in a matrix of pectin and hemicellulose. The cell wall undergoes greatest change at the meristem of the root or shoot tip, and these are the sites where the cell divide and expand. Young cells are connected by a thin primary cell wall which can be loosened and stretches to allow the subsequent cell elongation. This is possible only because of the nature of cellulose in a young cells. After the cell elongation ceases, the cell wall generally is thickened either by secretion of additional macromolecules into primary wall or more usually by formation of secondary cell wall, which is composed of several layers of cellulose. Experiment with the elongating root tip cells suggests that in primary wall, at least microtubule influence direction of cellulose deposition, suggesting a very critical role of cellulose in cell wall elongation. These cells have oriented bands or ring of microtubules located just under the plasma membrane, and these microtubules are transverse to the direction of elongation but parallel to many cellulose microfibrils in primary cell wall of elongated cells. Let us understand how does growth stop, that we can interpret what is a cessation of plant growth or cessation of cell enlargement. Once the elongation and expansion is complete, primary wall locks the cell into a particular shape. One component of this locking mechanism of a type 1 primary wall may be hydroxyproline rich glycoproteins. Hydroxyproline rich glycoproteins monomers accumulate early in a cell cycle and become insoluble during cell elongation and differentiation. But how these proteins are cross-linked in the wall either in a homo network or in a hetero network with other proteins is still unknown. Type 2 wall contain threonine rich protein with a sequences which are typical of hydroxyproline rich glycoprotein structures. Hydroxyproline rich glycoproteins as well as threonine-rich proteins are prevalent in vascular tissues, especially in a reinforced wall structures such as pericarp. However, much of the cross-linking function in type 2 walls probably rests with esterified or etherified phenolic acids, and formation of this cross-linkage accelerates the end of growth phase. Essentially, when a cell has reached to its final dimension, its wall is locked into a final hardened conformation. Molecules with specific role in growth cessation or inhibition of growth are thought to be exocytosed into cell wall, providing either substrates for cross-linkage reaction or the enzyme catalyze cross-linkage of pre-existing wall polymers. Let us summarize the topic. Irreversible cell expansion is a key step in plant growth and morphogenesis. Surface cell expansion of a plant cell wall may be highly localized, for example in growing root tip, or may be evenly distributed throughout the cell surface. These two processes occur in every plant cell of plant body.